Good day everyone. This is my virtual presentation for Malacca International Conference on Social Science, Science and Technology 2019. I am Sereni Ahmad Waked from School of Biology, Faculty of Applied Sciences, University Technologi Negeri Sembilan. My co-author is Nurul Safika Zahran. The title of this project is Antioxidant Activity of Vernonia cinerea, Peperomia pellucida and the combination of Vernonia cinerea and Peperomia pellucida. This is the abstract for this research project. Antioxidants are a wide range of substances or molecules that neutralize free radicals, and can inhibit damage to living cells, cited in Edom et al., 2015. The extracts were tested for their antioxidant activity in scavenging 1,1-diphenyl-2-picrylhydrazole, DPPH, radicals as cited in Ketsuan et al., 2017. The high antioxidant activity of the plant at low concentration indicates that it could be very useful for the treatment of ailments resulting from oxidative stress, cited by Onaka, 2016. Vernonia cinerea, also known as purple fleabane or little ironweed, is a non-native species which the origin is from India, as cited in Prabha, 2015. It is belonging to family Asteraceae. It is an annual herb and grown in tropical climate and semi-tropical climate areas as wasteland herb, and it can achieve its maximum growth to 1 meter height, cited by Khosr et al., 2011. Peperomia pellucida, also called silver bush or shiny bush, Onaka, 2016, belongs to the family Piperaceae. It is a herbaceous plant found in many South American and Asian countries. It has power to cure many diseases, as it was stated by the local community that the extracts of the plant was useful to treat bone aches and pains. The leaf was also used in headache, fever, eczema, abdominal pains and convulsions treatments, Khan et al., 2008. The objective of this research is to determine the antioxidant activity from 3 types of extracts from Vernonia cinerea, Peperomia pellucida and the combination of Vernonia cinerea and Peperomia pellucida by using DPPH free radical scavenging activity assay. For materials and methods, this research is conducted by following standard method in preparation of the extracts. Vernonia cinerea and Peperomia pellucida was obtained from UITM Kuala campus area. Each sample were washed and dried in oven at 40 degrees Celsius for 3 days. A blender is used to mill the samples into a fine powder and stored in airtight bottles at -4 degrees Celsius. Then, 25 grams sample of powder were weighed and separately extracted using 250 milliliters methanol to obtain final concentration of extract 100 mg/ml, Khosr et al., 2011. The suspension was filtered by using Whatman filter paper and was evaporated by using rotary evaporator. The extracts were kept at 4 degrees Celsius. The antioxidant activity of the extracts was determined by DPPH free radical scavenging assay. 25 ml of 8 mg/ml DPPH solution was added into each sample that has different concentration, 20, 40, 60, 80, 100 μg/ml, making the total volume of the mixture equal to 1 milliliter. The reaction mixture was incubated at room temperature and allowed to react for 30 minutes. The optical density was measured at 520 nanometers using UV vis spectrophotometer. BHT was used as a positive control. DPPH was expressed in terms of ascorbic acid equivalent antioxidant capacity, which was calculated based on its concentration of extract required to reduce DPPH radicals by 50%. The capability of melon peel extracts to scavenge the DPPH radical was calculated by using equation, scavenging activity, % equals 1-. Absorbance of sample at 520 nanometers, absorbance of control at 520 nanometers, x 100. IC50 value was determined from the plotted graph of scavenging activity versus the concentration of extracts, which is defined as the amount of antioxidant necessary to decrease the initial DPPH radical concentration by 50%. Triplicate measurements were carried out and their activity was calculated by the percentage of DPPH scavenged. For result and discussion, in DPPH assay as presented in the Table 1, it can be concluded that the scavenging activity of each extracts were moderately good. The highest percentage of DPPH inhibition of the extracts were recorded at their highest concentration, that was at 100 mg/ml concentration with Vernonia cinerea, 76.3%, combination of Vernonia cinerea and Peperomia pellucida, 71.21%, and Peperomia pellucida, 68.3%, respectively. Meanwhile, the DPPH inhibition presented by butylated hydroxytoluene, positive control, were 90.0% scavenging inhibition. For conclusion, the results in the present study showed that the extracts of Vernonia cinerea and Peperomia pellucida species showed scavenging effect towards DPPH free radical. Vernonia cinerea exhibits highest antioxidant activity compared to Peperomia pellucida and combined samples. More research is needed to establish the nutritional value of these extracts, especially in the fields of biochemical analysis that can contribute to human health. The authors are grateful to University Technology Mara for the facilities, support and financial aids during the research work. This is some references cited in this presentation. Thank you. For any question and comment, please email to serini@item.edu.my.